So just as a kind of a reiteration, this is what we said the SolCAP objectives were a few years ago. And so we wanted to make up to 10,000 markers and associate these markers with our carbohydrate genes, which are important in the processing industry. And so we've been able to make a SNP chip. There's 8,300 SNPs on there. So I think we're in that range. We wanted to collect standardized phenotypic data over multiple locations. Walter was reporting on that today, so I think we're moving forward. And let's not forget our tomato friends here. This project we're focusing today on the potato, but really I think what's helped driven this project forward is actually working with our colleagues in tomato breeding and genetics. I think having that expertise, I think, has helped us on the potato side. And I think the tomato people appreciate that they don't have tetraploids. So, and then we talked about having other populations that would be SNP genotyped. And so I showed that slide earlier that we have those different populations that have been submitted by the community. And so one of the questions I could put out to the people is, are there more populations out there that we need to consider that would be valuable to the community? Right now, the ones I have listed there, and even some others, we've got so much data to work with. We're going to keep ourselves quite busy. But I think this is, the bottom line right here is that we need to create this integrated breeder-focused resources, and so these databases and browsers that are developed. What Robin talked about, with her browser, I think is really critical. I kind of feel as a potato breeder, I got to be able to access that browser and use that information now that we have the genome sequence, and use that in our genetic analysis, and with the data, with the phenotypic data, with the SNP data available, these are going to be up there. As a breeder, how can I use that data? How is this going to help my breeding program move forward? So, what we feel that we've got these resources. We've got the cDNA libraries, the high throughput SNP analysis, the germplasm panels. We've got the SNP chips. We've been running these workshops. The whole point of the workshops is to help you feel more comfortable of where things are going, and we have the databases. And so what's going on in 2011? We've been SNP genotyping. All the obligations on the potato side for SolCAP have been done, and now we're just dealing with ones that people have submitted for their own purchased chips. We're working on the databases, and we're working on the publications. We're going to move to the QTL analyses, and we want to have a hands-on workshop to help people work through things. And also, we have the eextension.org site. Maybe when Dave Francis is talking, he can just take a minute and kind of update people on that. And so this is a place where also you should be able to find some of this information that might help you move forward. It should be a resource for the breeders, not just the potato breeding and tomato breeding community, but the breeders in general, where you can find information that would be helpful to you. So I think you can kind of capture it in this one line here, is that what's the outcome of SolCAP is a genome-wide set of markers and bioinformatic tools accessible by the breeders. So we're moving towards that accessibility. I think the tools are being developed, and I'm quite excited. And I'm just going to, this is my last slide here, and I'm kind of feeling like, okay, where does this all go with the, with this analysis? I remember we were saying, well, after we develop some SNPs, we'll convert them back to PCR-based agarose gel markers that you could use. Well, I think that's kind of silly. Really, the high throughput analysis is so much more cost effective. And so there's different platforms And so Illumina Bead Express, they have a platform, and you can generate, you can have a 48 SNP system, a 96, all the way up to 384 that you can analyze. You can multiplex, analyze at one time. That's what we want to do. We don't need to sequentially run all our markers that we need. We can run them in a few days, get our data. So you can see how they got it set up. If you want to buy 480 samples or almost 1,500 samples, what the cost is, right? So, and what this is, I think this is the column to focus on, is what's the price per sample to do the SNP genotyping. So as a breeder, I really don't know how many markers, when this SolCAP is all done, that we're going to think are important. And maybe as a breeder, you may think that yourself that you might say, oh, these are the ones that I think are important. Well, or we can do it collectively as a community. But if you say that we have 192 SNPs are important to us as a community, if we wanted to genotype progeny in our program, it's a little over $16 a sample for the reagents. There'll be a small, I think, genotyping cost, but the reagents are going to cost you there. So if you had 1,000 progeny that you wanted to SNP genotype, and so for a little over $16,000, you can do that. Well, and so the question is, will this help move your program forward? And I'm kind of feeling bullish about that there's going to be ways that we can do this. I think this is the exciting part, is where we're going to go with these markers. Can we create a breeder chip, or do we individually create our own breeder chips? But we have that option to do whatever we want. And our SNP chip was based on those 8,300, and you can actually, Robin mentioned that there's 69,000 out there, that fit the Infinium design. So you can go back to those and if you go into the browser, you can find them in there, and maybe those SNPs might be more valuable to you than the ones that we actually use. So this is how you have to start thinking. You can go back, the information's there, and how can you best use this? So I think we're entering kind of an exciting time, and I'm really curious. Like, we could sit around, drink a beer, and say, what's going to happen in the future with the SNPs? So I think, and I'll just finish with the acknowledgments. I really mentioned some of those people. This is really a teamwork to move forward with all this. I mentioned the people in Robin and my program that have worked on this, but it's really the team itself. I think if we didn't have our friends on the tomato side, I don't think we would also be there too. It's a great effort that's been going on. So I'll stop at this point and take a second set of questions, if there's any at this point. Paul.
When you say going through the individual SNPs to look for, are they segregating as 5, are they good SNPs, but you're seeing 5 categories in that. Is that really visually looking at every SNP?
Right. Okay, so Paul's asking if whether on those calls on each SNP, yes, it's actually an individual person's actually going SNP by SNP looking at that. So in other words, you get the Genome Studio auto call, but then what you want to do is just take a look at it and kind of agree, or you can change the parameters a little so that it's on what it's including in that call. Dave? So Dave Francis is pointing out that there is a cluster file, so you can go to that, and so you don't have to reinvent that and see what. So that. So Robin, the 3 cluster call is up on the site there, and the 5 cluster call will be up when it's finalized. And there's also, I think, one that's going to be going up for tomato also, right? So.
Online question, Dave?
We have an online question?
Yeah, from Gene Jacobson, New Zealand. Asking again about those the PMs. asking if the data is available, and especially the data not mapped in PM but assigned to linkage groups.
So so Jeannie Jacobs from New Zealand is asking whether those ones that haven't been assigned to chromosomes are going to be, so will the data be available on those, is what you're saying?
Right.
So we're in the process of writing the paper on that, and so I think that'll be coming out in the publications that we'll be submitting shortly.
Great, and she just typed in again to help improve the pseudomolecules, that comment.
Yeah, so I think the idea is to add that to the pseudomolecules. Raman, do you have a comment on that? Okay.
And
so that's where we are at this point.
Great, and one other quick one, you might have answered this one, but question is, are the mapping populations and maps going to be published so they can use the information?
So are the mapping populations maps going to be made available? So I expect that the maps will end up on SGN when, and SolCAP when as we finalize them there and submit our publications.
Great, that's it for now.
Oh, the populations, yeah, they can be put. They will be put on there. Well, the russet mapping population is on there right now, not the SNP data yet, but it will be. So Dave Francis is asking, can somebody get tubers for the populations? I think that you have to go to the individual person on that. And potatoes, that could be very difficult because of the challenge of maintaining a population and keeping the virus out of it. So that's going to come down to individual. I think we're talking about the mapping population and putting one of them into tissue culture. Richard, is your population in tissue culture? So, okay. So DM by RH is going into tissue culture, and we are going to move the DM by 84SD22 into tissue culture also.